Usually when we talk about biological survival, we kind of view it as this constant, desperate engine.
Right, yeah. It never stops.
Like you have to just keep consuming fuel, exchanging gases, maintaining your osmotic pressure, or the whole engine just seizes up.
Exactly. I mean, life demands this continuous forward momentum.
Yeah.
Just fighting entropy and maintaining a basic metabolic baseline, keeping the cellular machinery running that costs a massive amount of ATP.
Right. But, well, when you look at certain extremes in nature, that paradigm completely breaks down.
It really does.
We're looking at this biological landscape today where the engine doesn't violently seize up when you cut the fuel. It just glides into this perfect crystalline hold.
Yeah.
It is the absolute definition of hitting the biological pause button.
It forces us to completely reconsider the rules of metabolism. I mean, we are talking about a state that isn't just, slowing down.
Right.
It's a complete, fully reversible suspension of life processes.
And that is our target today. So welcome back to our ongoing deep dive series on the microscopic marvel that is the nematode worm.
Glad to be back.
Now, since you, the listener, are already a veteran of our C. elegans deep dives, we are entirely skipping the basic 101 biology today and just jumping straight into the deep end of the literature.
Straight into the good stuff.
Exactly. But really quickly, before we get into it, please take a second to follow, like, comment, and share this deep dive. It is the best way to help us grow the community.
Definitely.
Oh, and if you have your own published research that you'd like us to discuss in a future deep dive, make sure you reach out and contact Dr. Viren Chauhan. We love featuring your work.
We really do. Send those papers in.
Yes, please do. Okay, so our mission today revolves around this fascinating 2026 paper from Nature Communications.
Yeah, by Liu and colleagues.
Right, and it focuses on this phenomenon called suspended animation. So, okay, let's unpack this because we are looking at a shockingly simple way these worms hit that biological pause button. It is. It completely bypasses the extreme sledgehammer interventions that we used to think were completely necessary.
Yeah, for decades, the gold standard for inducing any kind of suspended animation in C. elegans, it involved forcing them into total oxygen deprivation.
Right, which sounds intense.
Very. Researchers would literally lock them in these specialized anoxia chambers, and they'd carefully scrub every single molecule of O2 from the environment to just force the worms to shut down.
Which, that requires heavy, cumbersome equipment.
Oh, absolutely.
You are actively forcing the organism into a corner until it has literally no choice but to stop moving.
Exactly. But Liu and the team, they discovered something totally different that they're calling LISA.
LISA.
Yeah, LISA, which stands for liquid-induced suspended animation.
And the trigger for LISA is just beautifully minimalistic.
It really is. It doesn't require stripping away oxygen. It doesn't require dropping the temperature to near freezing or anything like that.
Right.
All it requires is an isosmotic liquid. So something as standard as M9 laboratory buffer and a remarkably high population density.
It's crazy. The researchers actually described a, quote, standing procedure. You literally just let a massive crowd of worms settle at the bottom of a tube filled with M9 buffer.
That's it.
And within hours, they exhibit this complete cessation of all visible movement. And more importantly, all developmental progression just halts.
Yeah, the density of the population combined with that liquid environment, it creates this incredibly specific cue.
It kind of makes me think of a city's smart grid during a massive heat wave.
Oh, that's a good way to look at it.
Yeah, like instead of letting the entire power grid collapse under the load of millions of air conditioners running at once, the central software recognizes the dangerous density of the power draw.
Right.
And it automatically initiates rolling brownouts, like powering down non-essential sectors of the city just to preserve the core infrastructure until the load balances out again.
Exactly. Yeah, the worms are sensing the load and powering down.
But wait, I have to push back here for a second.
Okay, go for it.
We've covered the dauer phase on this show before. We know these worms have this famous survival mode they enter when things get crowded and food is scarce. So are we just looking at a rebranded dauer phase here?
That is a very vital distinction to make, actually. But no, LISA and dauer are mechanistically completely distinct.
Really?
Yeah. Dauer is a programmatic alternative larval stage. The worm makes a one-time developmental decision during its L1 stage to just deviate from normal growth.
Right.
And it becomes a dauer larva, undergoing these extreme morphological changes, like heavily thickening its cuticle.
Basically building a biological bomb shelter.
A permanent one, yeah. Until conditions improve, anyway. And that dauer decision is heavily reliant on specific genetic pathways.
Hmm.
Most notably, DAF22, which governs the synthesis of the specific pheromones that communicate overcrowding. But Lisa operates entirely outside of that system. Worms with mutations knocking out DAF22 saphigamuins that are physically incapable of forming dauer larvae, they enter Lysa without any issues at all.
Oh, wow. So the classic crowding pheromone isn't even the trigger.
Not at all. Furthermore, dauer is a one-time crossroads, right? Lisa can be induced across the organism's entire life cycle.
Wait, really? At any point?
Any point. You can take an L3 larva, an L4, or even a fully reproductive adult, drop them into this crowded liquid environment, and they just simply freeze in time.
That is incredible.
Yeah, and the researchers tracked these cell-specific reporters and literally watched ongoing processes halt mid-step.
Wow.
Like intestinal endomitosis, which is where the cells duplicate their DNA without actually dividing it, just stops. Even the terminal differentiation of the hypodermal seam cells, which are actively building the worm's outer skin layer, it just pauses mid-construction.
They just drop their microscopic hammers and walk out the job site.
Exactly. And what's fascinating here is that simply crowding them in this liquid flips a master switch. Yeah.
It points toward this highly sensitive quorum-like density sensor that we just don't fully understand yet.
Right, because it's not a chemical they're leaving behind.
Right. We know it isn't a stable, diffusible molecule in the water because if you extract the liquid from a densely packed pause tube and drop a few naive worms into it, they just keep swimming around normally.
Oh, I see. So they have to physically be in the crush of the crowd to actually feel the signal.
Precisely.
That implies some form of mechanosensation paired with, like, a short-lived chemical signal or something. That is incredible.
It really is.
But that leads to the next big mystery, right? We know the trigger, the smart grid sensing the load. But what is physically happening inside their biochemistry during this pause?
Yeah, that's the big question.
Because they aren't dead, but they clearly aren't running their normal engines either.
Well, the entire energy economy of the organism gets radically rewired. And to figure out exactly how, the team utilized liquid chromatography and gas chromatography paired with mass spectrometry. So LCMS and GCMS, basically taking a high-res snapshot of every single metabolite flowing through the worm system at a given second.
Exactly. It's a comprehensive biochemical audit, and the audit revealed a profound hypometabolic state.
Okay.
We see a steep drop in acetyl-CoA, which, as you know, is the primary carbon input for the TCA cycle.
Right.
Simultaneously, the ratio of NADH to NAD+ just plummets, signaling a massive deceleration in cellular respiration.
But the paper also noted an accumulation of specific metabolites, right?
Yeah.
Specifically succinate and lactate.
Yes.
If they're shutting down, why are those levels spiking?
Because you can never truly turn the engine completely off without dying.
Right.
You still have to maintain the resting membrane potential of your cells, which requires at least a trickle of ATP. When the primary aerobic pathways shut down, the worms shift to alternative anaerobic-like salvage pathways.
Just to keep that trickle flowing.
Exactly. The accumulation of lactate suggests they are relying heavily on basic glycolysis, while the spike in succinate indicates they might actually be running portions of the TCA cycle in reverse.
Oh, to act as an electron sink.
You got it. It is a brilliant bare minimum survival strategy to just keep the lights flickering enough to prevent cellular death.
Which actually brings up the live cell imaging from the paper, which is arguably the most visually striking part of the whole research.
Oh, the mitochondrial imaging.
Yes. They track the mitochondrial networks during Lysa. And as the metabolism drops, these normally long, beautifully fused tubular networks physically shatter.
Yeah, it's wild to see.
They break apart into these highly fragmented little vesicular spheres, accompanied by this sharp drop in mitochondrial calcium levels.
The mitochondrial network literally dismantles itself.
But, I mean, in almost any other context, mitochondrial fragmentation is a massive red flag.
Oh, absolutely.
If we see a cell's mitochondria fragmenting like that, we assume the cell is undergoing severe oxidative stress or, like, actively initiating apoptosis.
Programmed cell death.
Right. So why is this fragmented state suddenly a protective measure for a worm in stasis?
Well, context changes everything in biology. In a disease cell, fragmentation is often the first step of a catastrophic failure, usually followed by the release of cytochrome C and total cellular breakdown.
Right.
But the metabolomic data proves this isn't a passive failure. It is a highly coordinated biological lockdown.
So they're intentionally pulling the plug on the power grid.
Exactly. A fused, highly connected mitochondrial network is incredibly efficient at producing massive amounts of ATP, but it is also a massive liability when the system is paused.
Because of the free radicals.
Right. An active network continues to leak reactive oxygen species, ROS, and those free radicals will just tear the cell apart if the cellular defenses are lowered.
Wow.
So by deliberately fragmenting the mitochondria and dropping internal calcium, the worm actively suppresses its bioenergetic output. It stops the ROS leak entirely.
That makes so much sense.
It isn't a power plant collapsing from an earthquake. It is a power plant undergoing a carefully managed, controlled shutdown where all the control rods are fully inserted.
They essentially box up the volatile machinery so it doesn't accidentally burn the house down while the organism is asleep. That is remarkably elegant.
Very elegant.
But even with the power plant boxed up, just sitting in stasis for days has to create some microscopic wear and tear.
Oh, definitely.
Which brings us to how they manage cellular stress. And the RNA sequencing data revealed something incredibly counterintuitive here.
Yeah, the transcriptomics, so looking at which genes are actively being transcribed into RNA, it showed a massive upregulation of small heat shock proteins.
Specifically the HSP16 family, right?
Yeah, exactly.
And heat shock proteins are the classic molecular chaperones. Like when a cell gets stressed, normal proteins start to unfold and lose their 3D shape.
Right.
And the heat shock proteins physically bind to those unfolding proteins to stop them from clumping together into toxic aggregates.
That's their job.
But here's where it gets really interesting. The fluorescent reporters in this study proved that this massive explosion of HSP16 proteins doesn't happen during the suspended animation.
No, it doesn't.
It happens after they recover.
Yeah, the mRNA is transcribed and stockpiled during lysa, but the actual translation into functional proteins is strictly delayed until the worms are removed from the crowded liquid and actually start waking up.
It's exactly like a coastal city bracing for a Category 5 hurricane.
Okay, I like this analogy.
Like, you don't send your massive fleet of cleanup crews and cherry pickers out during the storm when the wind is literally tearing roofs off. That would be a disaster.
Right, they get destroyed.
Exactly. You stage the crews right at the edge of the city, engines idling, and the second the storm passes and the winds die down, you flood the streets with the cleanup crew to clear the debris.
That is a perfect way to describe it.
And in this scenario, the storm isn't the suspended animation itself. The storm is the terrifying shock of waking up.
Yes. In human medicine, we call it ischemia-reperfusion injury. Waking up from a severe hypometabolic state is incredibly dangerous.
Because of the sudden rush of oxygen.
Exactly. When you suddenly flood a dormant biological system with oxygen and ramp the energy production back up, you generate a massive spike of oxidative stress. Proteins misfold rapidly.
Right.
So if the worm isn't prepared, waking up will actually kill it faster than the stasis did.
Wow.
The delayed explosive HSP16 response perfectly buffers that reperfusion shock, preventing a proteostatic disaster the exact moment the metabolic engines reignite.
Surviving the wake-up call is literally just as hard as surviving the sleep.
It really is.
But the heat shock proteins were only part of the defensive strategy, right? The researchers also ran forward genetic screens to find out which specific cellular components were absolutely non-negotiable for surviving lysa.
And that screen led them straight to the autolysosomal pathway.
Which makes sense. I mean, if you were sitting in stasis, you still produce cellular junk, and you need a highly efficient recycling system to clear it out.
Right. And the live imaging of the lysosomes during lysa is just fascinating. Normally, lysosomes are somewhat spherical, distinct organelles. But during the stasis, they stretch out into these extensive, long tubular networks.
Really?
Yeah, tubulation is a very specific morphological signature. By stretching into tubes, they drastically increase their surface area.
Oh, to process more.
Exactly.
Yeah.
It heavily ramps up their autophagic flux, basically their capacity to capture and degrade cellular debris.
So they expand the recycling centers. And when the researchers intentionally broke that system, what happened?
The animals couldn't survive the pause. The team utilized genetic mutants with specific defects in autolysosomal trafficking.
Okay.
They targeted VPS15, which is a gene essential for forming the initial autophagosome, and VPS45, which governs the fusion of these vesicles.
Right, so breaking the infrastructure.
Exactly. In those mutants, the recycling infrastructure is broken. So when those worms were pushed into lysa, they suffered massive viability drops. The tubular remodeling of the lysosomes is a strict, non-negotiable requirement for prolonged survival.
They literally choke on their own microscopic debris without those expanded recycling centers.
Yes, exactly.
But through those genetic screens, they found one mutation that did the exact opposite, right? Breaking a specific gene actually made the worms dramatically better at surviving in suspended animation.
Yes. And it perfectly illustrates the delicate balance of stress responses.
Okay.
They isolated a mutant with a defect in a gene called DAF21. Now, DAF21 encodes the nematode ortholog of HSP90.
Right. And HSP90 is a master regulator of protein homeostasis across almost all eukaryotic life.
Exactly.
So wait, knocking out a master regulator of stress resilience makes them more resilient. I literally had to read that section of the paper twice.
It seems totally paradoxical until you look at the actual mechanism of HSP90. Under normal, non-stress conditions, HSP90 physically binds to and sequesters the heat shock transcription factor, HSF1.
Okay, so it holds it back.
Right. It essentially acts as a brake, preventing the cell from constantly mounting a massive, energetically expensive stress response when it isn't actually needed.
It holds the alarm system back.
Exactly. So by introducing a mutation in DAF21, the researchers essentially damaged that braking system. HSF1 was no longer fully sequestered. As a result, the stress response pathways became hypersensitive.
So the mutant worms were just permanently bracing for impact.
Precisely. These DAF21 mutants exhibited wildly exaggerated HSP16 expression upon recovering from lysa.
Wow.
And because they overprepared the cleanup crews, they showed significantly enhanced survival rates following extended periods of arrest compared to wild-type worms.
That is wild.
It proves that the wild-type organism calculates a very specific balance between energy conservation and stress preparation. Tipping the scale toward over-preparation buys the organism much more time in stasis, but it likely comes with heavy metabolic costs during their normal life.
That is deeply fascinating. The architecture of this pause is just incredible. I mean, they dismantle the mitochondria to stop ROS leaks, they stretch out the lysosomes into massive tubular recycling plants, and they heavily stockpile mRNA to deploy a massive protein cleanup crew the second oxygen returns.
It's a masterclass in survival.
It really is. But all of that preparation leads to the final critical hurdle of this paper. How does the worm actually know when it is safe to wake up?
Right, taking them out of the crowded liquid and putting them onto a standard low-density agar plate with food.
Yeah.
That initiates the recovery.
Yeah.
But returning to physiological normalcy isn't just a passive thawing out process. The exit from lysa is a highly regulated, deliberate neurobehavioral decision.
A decision. The worm's nervous system is actively debating the safety of the environment.
Exactly. And the researchers methodically screened mutant worms with specific neural and neurotransmitter defects to map the exact circuitry of that awakening.
What did they find?
What they uncovered is a literal tug-of-war within the worm's nervous system.
Like a battle between the alarm clock and the snooze button.
That's a great way to put it. The alarm clock is driven by a very specific neural circuit. The AFD sensory neurons act as the environmental sensors. They detect the shift away from the crowded liquid environment, and they immediately signal their downstream partners, the AIY interneurons.
And the AIY interneurons ring the bell. How does that signal actually propagate chemically?
Well, the single-cell RNA sequencing data highlighted that AIY neurons heavily express a gene called PDF1, which produces a neuropeptide called PDF.
Okay.
When the worm is placed in a favorable environment, calcium imaging visually confirmed that the AFD and AIY neurons fire aggressively.
Wow.
They release PDF, which binds to receptors that trigger an intracellular cascade relying on cyclic AMP-CAMP and protein kinase A, or PKA.
So that CAMP-PKA axis is the trigger.
Exactly. This signaling axis aggressively promotes behavioral awakening, overriding the stasis and engaging the motor circuits.
That is the alarm ringing loud and clear. But you mentioned a snooze button earlier.
Yes, the RIS neuron. In C. elegans neurobiology, the RIS neuron is famous for inducing and maintaining sleep-like quiescent states. During the exit from lysa, the RIS neuron acts in direct antagonism to the AFD-AIY circuit. It is actively releasing inhibitory signals, basically trying to suppress arousal and keep the worm in its protective stasis.
So the speed at which a worm wakes up is dictated entirely by which neuron wins that biochemical shouting match.
Exactly.
The AIY neuron is dumping PDF to wake the system up, and the RIS neuron is fighting to keep the system powered down.
And we know this definitively because of the ablation studies they did. If you use a laser to physically destroy the RIS neuron, the worms wake up prematurely and totally erratically. the snooze button is literally gone. Conversely, if you ablate the AFD or AIY neurons, the alarm clock is broken, and they exhibit a severe, prolonged delay in waking up, even if they are in a perfect environment.
This raises an important question, though.
Okay.
If we know the exact chemical wires of this alarm clock, specifically that CAMP cascade, can we hack it?
We absolutely can. And the researchers executed one of the most elegant experiments in the paper to prove it.
The optogenetic hacking.
Yes.
This was genuinely my favorite part of the entire methodology. They didn't just add a chemical to the dish. They genetically engineered a unique strain of worms to express a photoactivated adenylyl cyclase in their neurons.
It is a phenomenal technique. Optogenetics allows you to bypass environmental sensors entirely and just control intracellular biochemistry with photons of light.
So for our listeners who might not be deep into optogenetics, you essentially insert a protein that changes its physical shape when hit by a specific wavelength of light. In this case, when a photon of red light hits the protein, it undergoes a conformational change that exposes an active site. And that immediately starts converting cellular ATP into CAMP.
They installed a literal optical master switch.
The researchers have these engineered worms deep in the crowded, liquid-induced suspended animation. They don't change the environment. They don't give them food or take away the liquid. They just shine a red laser onto the dish.
It's so cool.
The light penetrates the tissue, hits those engineered proteins, and instantly floods the neurons with CAMP.
And the worms wake up immediately.
Precociously.
A laser snooze button override. They force the worms to wake up while still in the crowded liquid just by shining a light on them.
It is the definitive proof of the circuit. The intracellular CAMP-PKA signaling axis is the ultimate bottleneck for behavioral reactivation.
It's undeniable.
It doesn't matter what the environment is actually doing. If you artificially flood that pathway, the motor circuits engage, the metabolism tries to restart, and the pause is over. Okay, let's pull all of this together. We have an organism that can sense population density in a liquid environment and trigger a master pause button.
Right.
It dismantles its own mitochondria to prevent oxidative damage. It expands its lysosomal network into massive tubes to clear debris.
Yep.
It primes a massive stockpile of RNA to unleash a heat shock protein cleanup crew the second it wakes up. And it relies on a delicate neural tug-of-war driven by CAMP to safely reboot the system.
That's the summary.
So what does this all mean? Why are we dedicating an entire deep dive to the nap schedules of crowded nematodes?
Well, if we connect this to the bigger picture, the translational potential of this discovery is staggering.
It really is.
For decades, the medical community has viewed suspended animation as this nearly impossible physiological feat, something requiring freezing temperatures or total oxygen deprivation.
Right. Extreme interventions that cause massive collateral damage to complex tissues.
Exactly. You can't safely drop a human liver to near freezing for 3 days without destroying a significant portion of the cellular architecture.
It just doesn't work.
No. But Lisa provides a genetically tractable, fully reversible model of stasis triggered by a minimal environmental shift.
It's a game changer.
It proves that complex, multicellular life possesses a latent, inherent blueprint to safely pause and restart metabolism without requiring an extreme, damaging intervention.
It's like the software for suspended animation is already written into the DNA. We just didn't know the keystrokes to run the program.
That's a brilliant way to put it. And mapping those keystrokes, understanding the necessary delay in HSP16 translation, or the requirement for autolysosomal tubulation, or how to chemically override the neural alarm clock, it lays the groundwork for massive leaps in human medicine.
Absolutely.
The most immediate application is advanced organ preservation. Currently, the window for organ transplantation is brutally short, governed entirely by ischemia-reperfusion injury. If we can develop targeted therapeutics that induce a localized LISA-like hypometabolic state in a donor organ, boxing up its mitochondria and expanding its lysosomes, we could potentially extend the viability window from hours to days.
Which fundamentally rewrites the logistics of global organ networks.
Entirely.
Or consider acute trauma, right?
Yeah.
The golden hour in emergency medicine is dictated by how fast tissues die without oxygenated blood.
Yes.
If emergency responders could administer a therapeutic that flips a similar metabolic master switch, You could drop the cellular energy demand of a trauma patient to near zero.
Exactly.
You preserve their structural integrity, stop the ROS damage, and essentially pause the bleeding clock until they reach a surgical suite.
It takes the concept of metabolic suppression out of the realm of science fiction and grounds it in mapped, targetable biochemical pathways.
It does.
And yes, if we look far enough down the timeline, understanding how to safely dismantle and reassemble the cellular power grid is the foundational requirement for long-term controlled metabolic suppression in humans.
Which is the prerequisite for deep space travel.
Precisely.
It all comes back to that biological pause button. And honestly, it leaves me with one final lingering thought for all of you listening today.
What's that?
If a shift as remarkably simple as population density in an isosmotic liquid can trigger a master pause switch in a complex multicellular organism with a nervous system, what other invisible quorum-sensing cues might be hiding in plain sight?
Oh, that's a chilling thought.
Right. We are bathed in environmental signals we barely comprehend. What bizarre, undiscovered triggers are out there right now, just waiting to unlock dormant survival architectures in other animals?
Yeah.
Or maybe buried deep within our own mammalian biology.
It really forces us to ask how many hidden biological programs are written into our own genomes, simply waiting for an environmental signal we just haven't encountered yet.
It really does. Well, that wraps up our coverage of this phenomenal paper. Thank you all for joining us on this custom deep dive into the resilient architecture of C. elegans.
Thanks for listening, everyone.
And if you enjoyed this breakdown, please take a second to follow, like, comment, and share this deep dive with your fellow science enthusiasts. It is the absolute best way to help us grow this community.
Yes, please do.
And remember, if you have your own published research or a fascinating scientific frontier you want us to unpack in a future deep dive, reach out to Dr. Viren Chauhan directly. Until next time, keep questioning the world around you.